So first of all, thank you very much for the invitation. My name is Andrea Napolitano. I'm one of the medical oncologists working at the Marsden in the sarcoma unit. And I've been asked to present some of the data on the most recent and recently completed and ongoing clinical trials in GIST. And I know that patients are very well informed about the disease and they often know more than we do. But not knowing the level of expertise of the audience, I thought just to give a little bit of background information, because this is relevant to understand all the drugs that we're testing, and because as therapies are becoming more and more personalized, the molecular biology behind the disease is becoming more relevant also from a clinical perspective. So I'll try to share some of the molecular insights which are therapeutically relevant. So as you all know, GIST, gastrointestinal stromal tumors, they arise in the gastrointestinal tract for the most part, in the stomach, in the small intestine, in the rectal. Most commonly, they're gastric. The reason why I put this slide on is because it's important for us to recognize that different GIST arising in different locations, they have different chances of harboring specific mutations. We know that most GIST are driven in their growth by specific mutations, but it's important to keep in mind that these mutations are different in the different areas of the body where the GIST can arise. And these are pretty much the percentages of the different mutations that can be found in GIST patients. So about 95% of our patients have a GIST which carries one clear driver mutation. This is a mutation in a gene called KIT in about 70% of the cases, in another receptor tyrosine kinase gene called PDGFR-A in about 15% of the cases. And then there is a very large group of GIST patients which used to be called and is still called wild type, but actually in the last years there's been a characterization of this group at a quite granular level, and now we know that the so-called wild-type GIST actually encompass at least 4 or 5 different subtypes within their own. And this is all relevant because the drugs that we're going to discuss work in subsets of patients based on the molecular alteration that they carry. Next. Now we're going to go into the details, so please bear with me because this can be quite tedious, but again, I'm showing this slide because it has relevance for the drugs that we're developing. In the KIT gene, the mutations can be found in different parts of the gene and in different parts of the protein. So this is a graphic representation of how the protein looks like. There is a part which is outside the cell membrane, and then there is a part which is inside the cell. And within the portion of the receptor which is inside the cells, there are different domains, different regions. And the most common mutation in KIT is in the exon 11, which is in the juxtamembrane domain, which is the one just below the cellular membrane. Other mutations are found in the extracellular region, exon 9, and in the tyrosine kinase domains. And these are mutations in exon 13 and 14 and exon 17 and 18, which are down here. And importantly, PDGFR alpha mutation are usually, the most common one is called D842V in 80% of the cases. And this is a mutation which is important because it's resistant to the treatment with imatinib. So if we look at sensitivity to the drugs that are currently available, so imatinib, sunitinib, and regorafenib, we know that imatinib works quite well on KIT exon 9 and KIT exon 11 mutations, but it doesn't work when the patient carries mutation in exon 13, 14, 17, and 18. Sunitinib works well in exon 9, 11, and 13, 14, but doesn't work in 17 and 18. Regorafenib also has a differential pattern of response. Appreciate that this is sometimes not easy for the patient to appreciate, but again, working with this type of disease is starting to become really, really important to go into these details, because this is the reason why we're going to offer in the future one treatment over another is going to boil down to exactly this. Next. And another important thing to consider is that as the treatment goes on and as the disease progresses over time, the number of mutations and the type of mutations will increase over time. So every GIST usually starts with only a single mutation, which is what we call the driver mutation. But treatment exposure and disease progression will increase the number of mutations. We will have what we call secondary and tertiary mutations. And again, this is something that is already very well documented. For example, patients that have had at least imatinib have 40% of the cases that have at least 2 mutations, where only 15–16% have 2 mutations if they have not received imatinib. Likewise, patients with metastatic disease have 2 or more mutations in more than 47% of the cases, where they have 2 mutations only in 10% of the primary tumors. So we know that the number and type of mutation will increase over time and with the treatment. And lastly, just to finish this introduction, we know that one important factor to test the mutation is, of course, doing biopsies and seeing what the mutations are in the tissue. And this is what we usually do when we do a diagnosis, during the diagnostic process. But it's becoming more and more important also to assess the type of mutations in a less invasive way. And this can be done with what we call ctDNA. This is circulating tumor DNA. So basically, just with a blood sample, we are now able to identify the presence of all these mutations that we discussed previously in the blood. And this is important because it will give us an understanding of the whole range of mutations which might be different in different nodules. So if you do a biopsy only of one tumor, you will have an understanding of what the mutation is in that tumor, but you might miss other mutations which could be present in other tumor nodules. Whereas characterizing the expression of the mutations in the DNA which the cell shed in the bloodstream allows us to have a better understanding of the overall situation from a mutational perspective. Now, I checked the website clinicaltrials.gov, which is a website that lists all the clinical trials this morning, and there were 374 GIST studies, 197 completed, 19 phase 3 trials, which are the most advanced trials, the one which are seeking to change the standard of care, and 6 of these trials were open in the UK as well. So there are new trials opening very frequently, and there is renewed interest by companies with next generation drugs. I'm not going to cover all the trials. I'm going to give you a brief overview. So of course, if you have a favorite drug or a favorite trial, it may not be in the presentation. So we'll start with the advanced setting. So with the green symbol, these are the studies that have already been published and recently completed. The yellow are the ongoings, but I also have a couple of slides on studies that are going to be open shortly. So we can start with the Navigator trial, published a couple of years ago now. And this is a trial that was testing a drug called avapritinib, and it was specifically designed and created, this drug, to target the PDGFRA DA42V mutation. As you remember, this is the mutation which is imatinib insensitive. So this patient had no active treatment whatsoever. And this is called a waterfall plot. You can see basically on the axis, basically you can see how much the tumor shrunk whilst the patients were receiving the drug. And as you can see, almost 100% of the patients had some degree of shrinking, with a large proportion of patients having a degree of shrinkage of 40% or more. So the drug has been really, really effective in achieving the goal of controlling this particular disease subtype, which didn't have any treatment options so far. We mentioned NICE. the National Institute of Healthcare Excellence, which are the people that make the rules, and the drug is not recommended in the United Kingdom. And the reason why is that the company, when they applied, they received some feedbacks, and even the most plausible cost-effectiveness estimates were higher than the range normally considered cost-effective use of NHS resources. And at that point, after this feedback, the company decided to withdraw their submission. So the drug...
That's a long way of saying it's too expensive.
Yes. It's the way the NICE works is not too expensive in absolute terms, but it's too expensive based on the benefit. So paradoxically, if you have a drug which gives you a very long benefit, the NICE will have to pay for an expensive drug for a very long time. So they do these models in which they see how much it costs considering how long, on average, the drug will be used for. And this is how they come up with, is it cost effective or not? So here we have a drug which is really, really effective, which means that patients on this drug, even though it's a limited number of patients, will stay on this drug for many months or years. And the cost of the drug in that sense becomes too high.
It's the lifetime cost.
Exactly. Next. Now, the same drug was tested in a different trial in third line against the standard of care regorafenib in the Voyager trial. And as you can see, there is basically no difference in terms of survival curves. So these are Kaplan-Meier curves. You have the percentage of patients that have not progressed on one axis and the time on the other axis. So the two lines, the blue is the avapritinib, the new drug, and the red one is the regorafenib. They overlap. This was a study not specifically for patients with the D842V mutation. It was a broader study for all GIST patients. And in an unselected population, regorafenib is basically equivalent, if not marginally superior to avapritinib. So there's been no application for avapritinib in third line because based on the results of the study, it's not better than what we currently have. The Invictus studies, the other big player in recent years, the ripretinib. This is a study that was testing this other tyrosine kinase inhibitor, ripretinib, in fourth or more advanced line. The median progression-free survival, which is again the median time that the patient stays on the drug without progressing, was about 6 months. Next. And if we compare that with placebo, because this was a placebo-controlled study, in red you can see the curve for the placebo. Patients were progressing really, really quickly. You have a sudden drop of the red curve. Whereas patients receiving the ripretinib, which is this new TKI, were able to maintain a good response and survive without evidence of progression for, on average, 6 months. But we have patients, about 20-25% of patients that have been on the drug without evidence of progression for 1 year or even more. Again, we go to our NICE, and this is from the 23rd of March, so it's really recent. And the drug is not recommended. And again, the explanation is that it was not possible to work out if ripretinib is cost effective on the basis of the economic model presented by the company. This is an even more complicated issue because the drug is, the trial was testing the drug in fourth or more line. So you also had patients that had received even more treatment, but the application to the NICE was done only for patients in the fourth line. And the application at NICE was done saying that the patient would have been on the drug until evidence of radiologic disease progression. But in clinical practice, and you may know that, we tend to continue a drug even if the scan is showing a little bit of increase if the patient is experiencing clinical benefit, especially if it's the last line of treatment. So the way the drug would have been used in the NHS in the common practice would be slightly different because it would be restricted to the fourth line and potentially with what we call different stopping rules, because in clinical practice we use mostly clinical benefit as the stopping rule, especially in later lines of treatment, rather than just looking at the scan. And for this reason, at the moment, the drug is not recommended. Next. We have had an expanded access program of ripretinib in the UK, and the Marsden was the leading institute. So we have about 50 patients which have been treated with ripretinib outside of the clinical trial, just in the UK. And we recently presented these results at the ESMO Sarcoma Rare Cancers meeting in Lugano, and the results that we got in this real-life setting, which is different from the trial, is that the median survival is pretty much the same that was observed in the trial. We got 7 months compared to 6 months, so in line with what we saw in the study. And now we're discussing with the company as to whether we can try to, once these results are published, if we can try to use our real-life experience in the expanded access program to support the case for ripretinib approval by NICE. This is not, of course, easy, because especially when the company has to have discussions regarding the cost of the drug, these are not things that we can, really intervene. But I think that as a community, it's important to try to push to get these drugs on the market, because they're really showing, benefits for the patients. Now, the same drug which was effective in 4th line, so in very advanced patients, was tested in another trial, in INTRIGUE, in 2nd line against the standard of care, sunitinib. And similarly to what we observed before, when the drug is used in a particular setting or in a particular line, it may be really, really effective, but when it's then tested in a different setting, it may be less effective. And this was the case in an unselected population. The results that we observed with the ripretinib and with sunitinib were exactly the same. So based on these results, there was no application done, of course, to use ripretinib in 2nd line. Next. What was presented in January this year, which was really, really interesting and exciting, is this complicated slide, which I'll try to explain to you. So basically, we have a forest plot here. So you have favors ripretinib on one hand, favors sunitinib on the other hand, and then we have a bunch of subgroup of patients. And these are patients where in the ctDNA, that analysis of the bloods that we mentioned before, was available. So if you see the dot and the 2 bars, if they are all on one side, it means that in that particular subpopulation, one of the 2 drugs was better than the other. Whereas if the dot and the bars are right in the middle, it means that the 2 drugs in that subpopulation were basically the same. So what we're seeing here, and I want you to focus your attention on these highlighted bars, is that there are 2 clearly separate subpopulations, one in which sunitinib is much better than ripretinib, and another one in which ripretinib is much better than sunitinib. And these 2 subpopulations are defined by the presence in their ctDNA of those mutations that we were mentioning before. So patients that had an key exon 11 mutation together with an exon 13 or 14 mutation responded much better to sunitinib compared to ripretinib, whereas patients that had an exon 11 mutation plus an exon 17 or 18 mutation responded to ripretinib much better than sunitinib. And this for us was really, really interesting, because we're going towards personalization of earlier lines of treatment based on the mutation analysis, which is something that so far was not the case, because we have our 3 lines of treatment, but they're not really tailored based on the mutational status. And this is the first step towards personalizing treatment on the basis of the mutations which are found in the ctDNA. Now, this was what we call a sub-analysis of a different study. The original trial was not designed to prove that this is the case. So even though we got these results, no executive decision can be made on this analysis. Next. So what the company is now doing, they are designing a new trial which will open soon, specifically for patients which have these characteristics. So they have ctDNA with KIT exon 11 and KIT exon 17 and 18 mutations, comparing again ripretinib with the sunitinib, with the goal of showing that in that particular subpopulation, ripretinib is better than sunitinib in second line. Now the question, of course, we have is, okay, but how many patients are the ones that have this characteristic? It's about 20 to 25% of all patients. And this is because some patients which have a low burden of disease, they might not have enough ctDNA to be measured. And there are patients in which you do have ctDNA, but they don't have the right combination of mutations. But about 20-25% of patients will fall in this group. Next. And I'm glad that I put this slide, considering the audience. One of the trials which has already opened elsewhere and is going to open soon also in the UK as well is the PIC study, which is testing a combination in second line of sunitinib with another tyrosine kinase inhibitor, which is called bezuclastinib. So we've seen that the approach of one of the companies to select a specific subpopulation which is more likely to respond to a single agent, in that case ripretinib or sunitinib, but a different approach could be to combine two drugs which are differentially effective, meaning that we're combining, in this case, that we're trying to combine a drug, sunitinib, which is really effective in these exon 13, 14 mutations, with another drug, bezuclastinib, which is actually not really that much effective on that mutation, but is effective on the mutations which are not covered by sunitinib. So in this case, the approach is to combine two different drugs to try to cover all the mutations that can be found after progression to imatinib. And this is a trial that is currently open. We're all going to open soon at the Marsden as well. And the goal is to see which one of these approaches is going to be the winning one. Other trials, there are, again, as I mentioned, hundreds of new trials, but the basic idea is that there are a combination of new drugs with old tyrosine kinase inhibitors. For example, as we saw, the combination of a new tyrosine kinase inhibitor with sunitinib, or the combination of pimitespib, which is another new molecule, with imatinib, which is an old tyrosine kinase inhibitor. But there are also new drugs. And these are two of the most promising drugs which are currently in development. And these are novel tyrosine kinase inhibitor, which in vitro and in animals have shown to be more effective even than imatinib itself in inhibiting all the mutations. So they are more potent, and they cover more mutations compared to imatinib. So in the coming years, we're really excited to see whether these new tyrosine kinase may actually finally change also the first line of treatment, if they can prove to be more effective than imatinib in patients as well. Next. So this was for the advanced setting, which is of course where most of the research is conducted, but I just wanted to mention one single study in the adjuvant setting as well. As you know, we've used over the course of the years many tools to try to understand which are the patients that have a higher risk of GIST recurrence after surgery. The variables that have been used to assess which patients are high risk or low risk have been pretty much the same over the course of the year. So tumor size, tumor site, the mitotic index, so the presence of mitosis, and more recently in the classifications from 2008 and 2012, also the presence of tumor rupture. Next. And these are in the 2012 patients by Joensuu. We have now developed these charts which can basically, based on these variables, they will tell us which are the patients that have a higher risk of GIST recurrence. And as we know, the standard of care for patients with a high risk of recurrence is imatinib and KIT sensitive and imatinib sensitive mutation. The standard of care is to offer imatinib for 3 years. But we know that there are patients whose risk is close to 80, 90, 100%. So next, there is Scandinavia trial, which is open also in the UK and now has completed the recruitment, testing longer imatinib in patients with a very high risk of recurrence in the adjuvant setting. This means after the patients have had the surgery, and the drugs are given for a fixed amount of time to try to delay or prevent the recurrence. And the standard of care is 3 years, but now we're testing a duration of 5 years to see if extending the treatment is going to lead to benefit for our patients. So the take-home message is that we have had 2 recently completed positive trials. Again, the avapritinib in the D842V population and the ripretinib in 4th line. However, no new NICE approval. And the question is what we can do better as a community of doctors and patients to try to push for these drugs to be available for our patients. The second take-home message is that there are promising upcoming and ongoing trials in the advanced setting, so there is hope for new treatment options. It may take some time because these are the times of the research, but we're really excited because there is renewed interest in developing new drugs targeting KIT and PDGF-alpha. And the 3 versus 5 years in the adjuvant setting has completed recruitment, and we're waiting the results to see if this can improve outcomes in population at high risk of relapse. Thank you.